Welcome back to Sri Lankan Life Sciences Academy. In today's video, we dive into lesson 13 from Unit 4, Statistics and Ecology. Let's explore how statistics help us make sense of ecological data and identify real patterns in nature. By the end of this lesson, you should be able to describe the basic statistical concepts used in ecological studies and apply those statistical methods to analyze and interpret ecological data. In ecology, patterns in data can appear due to biological relationships or simply by chance. Statistical tests help us determine which is which. Some of the most commonly used statistical tests in ecology are Spearman rank correlation coefficient, student t-test, chi-square test. Let's learn how they work. Before collecting data, it's important to have a clear hypothesis, what you aim to prove or disprove. We also define a null hypothesis or H0, which assumes that any observed differences are due to chance, not actual effects. Once we calculate the observed value using a statistical test, we compare it to a critical value from the reference table. If the observed value exceeds the critical value, the result is statistically significant. It likely didn't happen by chance. This bell curve helps us visualize what significant means. If a value falls within the critical region for p is less than 0.05, we are 95% confident that the relationship or difference is real. So, what happens after analysis? If the result isn't significant, we accept the null hypothesis. If the result is significant, we reject the null hypothesis and conclude there is a real effect. Spearman rank is used to measure the strength and direction of a relationship between two variables. It gives a value +1 for perfect positive correlation, -1 for perfect negative correlation, 0 for no correlation. Let's look at some null hypotheses for Spearman test. There's no correlation between algae depth and photosynthesis. There's no correlation between study hours and exam scores. There's no correlation between population density and genetic diversity. Here's how to calculate RS. Rank both variables. Find the difference between ranks. Square those differences. Add them up and plug into the formula. Simple steps, big insights. Now interpret your results. If RS is 0, there's no correlation. Accept null hypothesis. If RS is close to +1 or -1, reject the null hypothesis. There's a strong correlation. This table helps you compare your RS value with critical values based on sample size to determine significance. Let's try an example. Marine algae were ranked by photosynthesis rate at different depths. Can we find a correlation between depth and photosynthetic efficiency? We calculate the rank differences, square them, sum them, and apply the formula to get RS. In this case, RS is equal to 0.3. This shows a weak positive correlation, and since it's below the critical value, the result is not statistically significant. Students' t-test help compare the means of two groups to see if a difference is significant. This is useful when comparing things like plant height or growth rates. The data should be follow a normal distribution. The test checks if the difference between means is real or due to chance. Example null hypothesis. No significant difference in height between males and female students. No significant change in student scores before and after tutoring. No difference in drug effectiveness. Calculating t-value. You will need the mean and variance of both groups, then plug into the formula to find the t-value. Compare the t-value with the critical value using the degree of freedom and p-value. If p is less than 0.05, the result is significant. Reject the null. Here's a real-world study comparing plant growth in sandy versus clay soil. Do different soil types affect plant growth? Let's apply the t-test. The chi-square test checks whether observed results match expected outcomes. It's great for categorical data like species distribution in habitats. The formula is simple. O observed values, E expected values, sum all O minus E squares and divide it from E to get chi-square. Example hypothesis. There is no association between the presence of species A and species B in quadrats. Their distributions are independent. To apply the test, use the data table, calculate expected values, use the formula to find the observed values. Now, calculate degrees of freedom equal number of categories minus 1. Find your critical value for p is less than 0.05 and compare with your observed chi-squares. Let's determine if a plant species is equally distributed across forest, grassland and wetland habitats. We'll use the chi-square test. Step 1. State the null hypothesis. Step 2. Calculate expected frequencies assuming equal distribution. Use the formula to calculate the chi-square statistic. Degree of freedoms equal 3 minus 1 equal 2. For degree of freedom 2 and p is less than 0.05, the critical value is 5.99. If the chi-square value is greater than the critical value, we reject the null hypothesis. That means the plant is not equally distributed. Habitat type matters. Thank you for joining us today. Now you have learned how to apply 3 powerful statistical tools in ecology. Use them wisely to discover the hidden patterns in nature. Don't forget to like, subscribe and stay tuned for more lessons here at Sri Lankan Life Sciences Academy.